In lecture 4, we're going to go over cells. So for this chapter, we're going to cover sections 1 through 5. So before we talk about the different types of cells, let's talk about cell theory. Cell theory states that a cell is the structural and functional unit of life. So meaning that all organisms contain cells, and how well these organisms functions depends on individual or combined activities of those cells. So the function of the cell is dictated by the structure of the cell. So for instance, biochemical functions of cells are dictated by the shape of the cell and by specific subcellular structures. And it also states that cells arise from pre-existing cells. As far as cell diversity, there's about 250 different types of human cells, and they differ in size, shape, and subcellular components. And these differences lead to these cells having different functions. So for example, we have the erythrocytes, which are the red blood cells. We have epithelial cells, which line organs. We have muscle cells, like skeletal and smooth muscle. These cells are obviously involved in muscle contraction. We have the adipocytes, like these fat cells that store nutrients. They also provide some kind of cushion. The, the macrophages, which are immune cells. And then we have like the reproductive cells, like sperm and nerve cells. So all cells have some common structures and functions. And human cells have three basic parts. So there's a plasma membrane, also known as the cell membrane. This is the flexible outer boundary that separates the inside of the cell from the environment. Then we have the cytoplasm, which is the fluid portion of the cell. So the cytoplasm is enclosed by the cell membrane, and it contains the fluid, which is the cytosol. Then there's the nucleus, which is the DNA containing portion of the cell. Okay, so here just a diagram of a cell. So we're mainly going to focus on the plasma membrane or the cell membrane here. And we're going to look at the different types of proteins that you can find on the plasma membrane. Proteins and some lipids. So the plasma membrane acts as an active barrier. It separates the intracellular fluid from the extracellular fluid. So intracellular fluid would be here, surrounding the organelles, like the mitochondria, the ribosomes. Extracellular fluid would be the fluid outside of the cell, so it would be somewhere out here. So the cell membrane controls uh, what enters and what leaves the cell. So structures that you can find on the cell membrane would be like the membrane lipids. These form a flexible bilayer. So this is the phospholipid bilayer that I've already introduced. There's also membrane proteins that float through the fluid membrane, and this results in a constantly changing patterns. So that's why it's referred to as the fluid mosaic pattern. So pieces that are in constant motion. And then there's also these surface sugars, which are called glycocalyx. Glycocalyx, these function as barriers for, it could be macromolecules or things like bacteria. So it protects the cell membrane. And they also play a role in cell-to-cell recognition. So they serve as identity markers. There's also membrane structures that hold cells together through cell junctions. So here you can see the model uh, of a cell membrane. So we have the membrane proteins and the glycocalyx. So glycocalyx is the it's a structure that involves a sugar and a protein. And you can see the phospholipid bilayer, the two layers of phospholipids. Okay, so all of these contribute to forming a barrier that allows the cell membrane to keep things or some things out of out of the inside of the cell. So these phospholipids, they have two components to them. There's the polar head and the non-polar tail. The head portion is the one that's hydrophilic. So this part likes to be near water. And the non-polar tail is the one that's hydrophobic, the one that does not want to be in water. So this phospholipid bilayer prevents a lot of the things from going into the cell, inside of the cell. So anything that's either charged or any molecule that's too big will not be able to will not cross the phospholipid bilayer. Some of the things that can pass would be things like water, because even though water is a polar molecule, it's so small that it does slip through the phospholipid bilayer. The membrane proteins, they have different functions. So they are involved in transport, so transporting molecules from outside of the cell to the inside. They serve as receptors for signal transduction. They have enzymatic activity, so they could be enzymes that are activated by molecules. They can function in cell-to-cell recognition. Uh, they could serve as attachment to the cytoskeleton and the extracellular matrix, and they can be involved in cell-to-cell joining. The cell junctions, um, this is what allows the cells to be bound to each other. So the three types are tight junctions. A tight junction is an impermeable junction that prevents the fluids or solutes from moving in between cells. So in this here you can see the tight junction. So it prevents things from going between the cells. So it forces those things to come across the cell. The desmosomes, these are anchoring junctions, so they contribute to uh, a network of you could say wires that reduce the chance of tearing due to physical forces like pulling forces. So desmosomes, you would find these in tissues like cardiac tissue or the epithelium that's lining the esophagus. So areas where that could be prone to tearing, these desmosomes prevent that. So here you can see what it would look like for the desmosome. So it creates a network that holds these cells together. So it prevents them from being torn apart. Gap junctions are formed tunnels that will allow ions, simple sugars, and other small molecules to pass from one cell to the other. So here you can see these gap junctions. So these are little tunnels from that join the two cells. So it allows for things to move from one cell to the next. So some of the ways that things can move into the cell include things like passive membrane transport. So this type of transport, it says passive because it requires no energy. Some of the examples include simple diffusion. So that would be, for example, when water diffuses across the cell membrane, it does so by simple diffusion because no energy is being used. Water is just moving from an area of high water concentration to an area of lower water concentration. But water is it's an exception because typically polar molecules, so molecules that have a charge, cannot cross via simple diffusion. But because water is it's small, it's a small molecule, it's able to slip through. The other things that could cross uh, by simple diffusion would be lipid solubles or nonpolar substances. For facilitated diffusion, this does allow for larger molecules, um, also non-lipid soluble molecules, and polar molecules. So polar molecules can cross, the larger molecules can cross, and the non-lipid molecules can also cross using facilitated diffusion. And they're able to do this because of the carrier molecules. So in this situation, the molecules are still traveling uh, down their concentration gradient. So they're going from an area of high solute concentration to an area of lower solute concentration. So that's why it's called moving down a concentration gradient. The only difference here is that these use carrier molecules. Osmosis, this is the movement of water, okay, from an area of high water concentration to an area of lower water concentration. So in this diagram, you can see this diagram illustrates a concentration gradient. So when a dye pellet is added to a container containing water, immediately surrounding the pellet, there's going to be a higher concentration of dye. So that's an example of a concentration gradient because you have high concentration of the dye and then there's lower concentration away from the pellet. So due to random thermal motion, which is kinetic energy, this makes the dye molecules move and collide constantly. So this is going to result in the dye molecules to move down their concentration gradient. So the concentration is going to be higher around here, but just due to random thermal motion, it's going to cause those dye molecules to move down their concentration gradient. So they'll move from a high concentration to a lower concentration until eventually it's evenly distributed, and then there's no more net movement of dye. So facilitated diffusion, like I mentioned, it allows for uh, certain hydrophobic molecules like glucose and amino acids to be transported passively down their concentration gradient. So from lab, if you remember, there was a question that was related to this. The question was concerning dextrose, which is I mentioned you can just consider that glucose. So glucose is able to be absorbed by the red blood cells. It's able to be absorbed because of these carrier-mediated structures or proteins that are located on the cell membrane. So it allows for it to take in those glucose molecules by binding to the protein carriers. There's the other type are known as channel-mediated facilitated diffusion. So this is when substances move through water-filled channels. So a lot of this is how the small intestine absorbs a lot of those substances. Osmolarity is the measure of it's the concentration of the total number of solute particles in a sol in the solvent. And water concentration varies with the number of solute particles because the solute particles will displace the water molecules. So for example, if you have a solution where the solute concentration is higher inside of the cell versus outside of the cell, then the water inside of the cell will be at a lower concentration, and the water outside of the cell will be at a higher concentration. Okay? So the water molecules are always going to follow the higher concentration. So here, so water moves by osmosis from areas of low solute, which means that it's going to have high water concentration to high areas of solute and low water concentration. So they're inversely related because the solute particles will displace the water molecules. So here in this diagram, you can see how the water moves. So if you have a freely permeable membrane, that means that things can move back and forth. If the solute concentration is higher here than it is here, then the water will move from an area of low solute concentration to the area of high solute concentration. But I mentioned they're inversely related because here, if the solute concentration is higher here than it is here, that means that the water concentration is going to be lower here than it is here. But this is freely movable, so it's not a semi-permeable membrane. So here, the solutes can move back and forth. So in this case, the water would just move from this side to this side. If you have uh, a membrane that's permeable to water but impermeable to solutes, so meaning that the red dots, the solutes can't cross, and you have two different types of concentrations or two amounts of solutes, high solute here, low solute here, that means that only water will move. So in this case, only the water moves from an area of high water concentration to an area of low water concentration. And in this case, it would just increase the volume on the side with the higher solutes. Tonicity, that's the ability of a solution to change the shape of cells by altering the cell's internal water volume. An isotonic solution would have the same osmolarity as inside of the cell, so the water volume would remain unchanged. The hypertonic solution has a higher osmolarity than inside of the cell, so it has a higher concentration of solutes outside versus inside of the cell. So in this case, the water flow, the water is going to flow out of the cell, and that's going to result in cell shrinking, also known as crenation. A hypotonic solution has lower osmolarity than inside of the cell, meaning lower solutes or lower solute concentration outside of the cell. So in this case, the water flows into the cell, resulting in cell swelling. And if it continues to flow into the cell, then it could lead to cell lysis. Osmolarity is equal to molarity times the number of ions. So for example, sodium chloride, that's one particle, but that ionizes to sodium and chloride, which is two particles. So from your lab, if you remember, you had to calculate or you were dividing the percentage of sodium chloride over the molar mass, which was 58. Once you did when you did that and you got an answer, you had to multiply that answer times two, and that's to account for the two particles of sodium and chloride. But this only applies to to molecules uh, that have ionic bonds, so not all of them. The two major active membrane transport processes are active transport and vesicular transport. So both of these require ATP to move the solutes across the plasma membrane. And it could be for several reasons. Either the solute is too large for the channels, or the solute is not a lipid soluble, or the solute is not able to move down the concentration gradient. So there's two types of active transport. There's primary active transport, which requires energy, And this comes directly from ATP hydrolysis. And then there's secondary active transport that also requires energy, and this is obtained indirectly from ionic gradients that are created by the active transport. In primary active transport, the energy from hydrolysis of ATP causes a change in the shape of the transport protein. And the change in shape is going to cause the solutes to bind to the protein, or the solutes that bind to the protein to be pumped across the membrane. So an example of this would be the sodium potassium pump. The sodium potassium pump, this is one that I'm pretty sure I went over in one of the other videos, or I might have covered it briefly. It's basically, it's an enzyme. So it's the sodium potassium ATPase, and this pumps sodium out of the cell and potassium back into the cell. And these are located on the plasma membranes. Okay. These are important functions for two types of tissues, so muscle tissues and nervous tissues. So starting here with the primary active transport, this is the one that's going to create uh, a, a, so, a gradient, a concentration gradient for sodium. So it starts off by pumping the sodium out of the cell. So it'll pump 3 sodium molecules out of the cell, and it'll take in 2 potassium molecules. Then we have the secondary active transport, okay, which, because sodium is now at a greater concentration outside of the cell than it is inside of the cell, it's going to move down its concentration gradient. So it'll go from high to low, and it'll want to it's going to cross this co-transporter. As it does that, it's going to bring in those glucose molecules with it. Okay. So this is the way that these two work together. The sodium potassium pump creates that gradient, that gradient that allows for sodium to move down that gradient. And in the process, it brings in glucose with it. For vesicular transport, these processes include endocytosis, which transports into the cell, and there's different types of that. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. There's exocytosis, which is going to transport out of the cell. Transcytosis, which transports into the cell, then across, and then out. And then there's the vesicular trafficking, which transports from one area of the cell to another. Endocytosis, these involve receptors. So because they involve receptors, it can be a selective process because the substance has to be able to bind to those receptors. Once the vesicle is pulled into the cell, it can either fuse with a lysosome or it can undergo transcytosis. So as you can see here, the vesicle is pulling the substance into the cell. Once it's inside of the cell, it's either going to fuse with a lysosome, like it's shown here, for the substance to be digested, or it's going to go to the opposite side of the cell. So that would be considered transcytosis to release the contents. Phagocytosis, phago meaning eating, and then cyto or cyte means cell, so cell eating. These use membrane projections that are called pseudopods to form around solid particles that are going to be engulfed. And then it forms a vesicle which is pulled into the cell. So example of this would be like that cells that do this would be the white blood cells. Then we have pinocytosis, which is another type of endocytosis. This is referred to as cell drinking. Here, the plasma membrane infolds and it brings the extracellular fluid into the cell. Then there's receptor-mediated endocytosis, which involves endocytosis and transcytosis of specific molecules. For example, there's enzymes, there's low-density lipoproteins, insulin. Could also be viruses or cholera toxins. They can also be taken into the cell in this manner. Exocytosis is where the material is taken out of the cell. So here the substance being ejected is considered, it's enclosed in a secretory vesicle. And what you have is you have a protein vesicle called the V snare, and this is going to bind to a target T snare proteins on the membrane. Some of the substances that are in exocytosed would be things like hormones, neurotransmitters, and cellular waste could be another example. So here you can see the T snare on the cell membrane, the V snare on the vesicle. So the membrane-bound vesicle is going to migrate to the plasma membrane. There, the proteins of the vesicle surface are going to bind with the T snares of the plasma membrane. And then the vesicle and the plasma membrane are going to fuse and the pore is going to open and then the contents are going to be released to the cell, to the outside of the cell. Resting membrane potential, this is the electrical potential or electrical potential energy that's produced by oppositely charged particles across a plasma membrane. The difference in electrical charge between the 2 points is referred to as voltage, and the voltage only occurs on the membrane surface. The rest of the cell and the extracellular fluid are going to be neutral. And as far as ranges, that's going to range between -50 to 100 millivolts in different cells. The way this starts is by potassium diffusing out of the cell through potassium leakage channels, moving down its concentration gradient. Then potassium is going to be pulled back in because when potassium leaves the cell, because since it's a positive molecule, when it leaves the cell, it's going to make it more negative. So potassium is going to leave the cell. That's going to make the inside of the membrane more negative. And then eventually potassium will be pulled back in by the more negative interior. Okay, so it moves. So just to review, potassium moves down its concentration gradient. So it goes from the inside of the cell out to the outside of the cell. And by doing that, it's making the inside of the cell more negative at the cell membrane. And then that will draw potassium back in because it would want to, because of the electrical gradient. And then when the drive for potassium to leave the cell is balanced by its drive to stay, then resting membrane potential is established. And that would be around -90 millivolts. Sodium can also affect the resting membrane potential because sodium is also attracted to the inside of the cell. So sodium being a positive molecule, it'll also be attracted to the negative charge. So if sodium enters the cell, it's going to bring up that resting membrane potential to -70 millivolts. But the membrane is more permeable to potassium than sodium. So potassium is the primary influence of resting membrane potential. So here, so let's just go over this one more time. So we have potassium being at a higher concentration inside of the cell. So it's going to move down its concentration gradient. When it does that, it'll leave the cell. Once there's enough potassium out here, this is going to turn negative because potassium has a positive charge. And when the cell loses molecules with positive charges, it's going to make the inside of that membrane more negative. So potassium is out here, but now that this is negative, it's going to draw potassium back in because of the electrical gradient. So it's going to move potassium back into the cell. And once the movement of potassium into the cell equals the movement of potassium leaving the cell, then at that point, um, resting membrane potential has been established. Okay, so that would be it for cells. Um, and we'll continue the next chapter on the next video.